quite interesting contrast to the last talk, in that I'll also be talking about agents, but very different types of agents. These are AI agents that we build, in particular agents that are served as scientists to help us to make scientific discoveries. So, usually when we think about AI, right, in science, we're thinking about AI as a tool, right, a tool for solving usually a pretty specific and often narrow scientific problem. Right, so an example of this would be like, for the problem of predicting protein structures, right, then AlphaFold is a very powerful tool that can solve that very specific problem. Right? But often, that's just a piece of the research, right? There's often a lot of other things that goes on beyond just predicting the structures. You have to figure out what data goes in, how to interpret the output of AlphaFold, right? So, but it's still a very useful tool. And I think what's really exciting is that especially in the last year and 2 years, right, there's sort of an interesting paradigm shift where now the models and these AI models are becoming more flexible, more powerful, right? They'll have these AI agents now that they themselves can use different tools like AlphaFold, but they can also talk to other AI models, talk to human researchers, right? So we're really in this interesting possibility of thinking about AI not just as a tool, but almost like as a teammate that can help us to make sort of interesting scientific discoveries and also help us throughout the scientific process. So that's sort of the idea and the context with which I want to explore this idea of the AI agents today, of these AI scientists. And in particular, I would want to sort of give 2 concrete examples of these AI scientists that we've been working on developing recently. So the first example is what we call the virtual lab. It's basically a lab of AI scientists, agents, that work together to make some quite interesting discoveries that we experimentally validate. And in the second example, second half, I'll tell you about sort of these AI agents that we built to actually help reviewers, right, which we deployed as a part of the recent iClear conference, where this agent helped to provide assistance to about 20,000 reviews submitted to iClear. Okay, and if you have questions at any point, also just feel free to interrupt me. I know this is a quite diverse audience, people from different backgrounds. So first, I want to explain a bit of this virtual lab. So the idea of the virtual lab is that we designed this to be sort of a platform where you have a team of interdisciplinary AI scientist agents, right, that work together and also with human researchers to sort of actually solve some quite challenging open-ended research projects. Right, so in the virtual lab, we designed there's actually like an AI professor agent, right? And under the AI professor is a bunch of AI sub-agents. So think of them as basically students, agents, right, with different expertise. So there could be like an AI computer scientist, AI mathematician, AI chemist, physicist. And they work together with the AI professor to tackle these quite challenging problems. And just, I'll tell you how the lab works, but also just to illustrate, make it very concrete, I'll give you an example of how the AI agents, this virtual lab, actually designed new nanobodies, right, which are sort of like little small proteins that can bind quite well to the recent COVID variants, right? So we can potentially serve as like building blocks for potential vaccines and therapeutics. And we've experimentally validated these nanobodies. Okay, so we set up the virtual lab, right, to be maximally flexible and quite interactive. So the way that we, the human researchers, that we interact with these, each of these AI agents is that with the virtual lab is primarily we talk to the AI professor, right? So like we can tell the AI professor, for example, we're interested in designing binders, right, to COVID variants, or we're interested in solving this physics problem, right? And then it's actually the job of the AI professor to automatically create and then to train the sub-agents that have the relevant expertise. So in this particular case of designing binders for COVID, right, so the AI professor would decided, we also call this the AI PI, decides that it's useful to have like an immunologist, useful to have a machine learning specialist, and also a computational biologist, right? So then it's actually the job of the AI professor, PI, to create and to train those sub-agents. So under the hood, right, so each of these agents is basically our large language model. In this case, we use GPT-4o, but we could use other models. And we actually have a sort of a recipe that we call the virtual lab school, where these language models become and develop their expertise in each of these domains through like a process of self-teaching and self-improvement. Right, so I'll actually come back to that in a few slides. But for now, you can imagine that the professor actually sort of creates these agents, really trains the agents, And then we also create what we call like a scientific critic, essentially like a kind of a reviewer agent that participates in all of these virtual lab meetings. So once the virtual lab is created, right, so then the way that they start to tackle these research problems is through two types of meetings. So there will be like virtual lab group meetings where all of the agents meet together and sort of participate in the discussion. And then individual meetings where the professor may meet with one of the agents to solve a specific subtask.
Excuse me.
Yeah.
Do you have any type of measurement and quantification of the time points at different scales? What is the time that takes time for that process?
Yeah. So I'll give some specific numbers, but each of these virtual lab meetings is usually quite fast. It's usually on the orders of like a few seconds to less than a minute.
But the whole process, do you have at the different
scales? So I'll get to that maybe toward the end after I show the actual experiments. But the longest part of the process is actually for the humans to do the experimental validations, right? So that's when we do the experimental validation. That took about 6 weeks. But the virtual lab part is on the orders of days.
I see.
Yeah.
Oh, I'm just wondering, do these like cute little images correspond to roles that you pre-assigned, or do these just naturally occur through the experiment, or do you pre-assign?
So good question. So we don't tell the professor agent that you have to have an immunologist, right, or a computational biologist. So the human researcher, we describe to the professor agent, here's our high-level goal. We want to design binders to COVID. And then it's actually the job of the PI agent then to automatically create the sub-agents that have the relevant goals, right? So it's not pre-assigned. It's basically the job of the PI to actually create each of these roles and then to train those sub-agents to have those expertise. So if you have a different problem, right, maybe your problem's in child development, right, or in quantum physics, right, then the professor will actually create a different set of agents.
Why can't the professor or the PI be smart enough to know everything?
So it's a good question. So we've actually I'll show some examples where we actually do ablation experiments to see that there's actually a lot of benefits to having multi-agent interactions rather than having a single agent or like single Uber agent to do these problems. But the short answer is that actually you see a lot of interesting debates and disagreements that happens across these different roles, which actually leads to much deeper, more in-depth scientific reasoning.
Yeah. To what extent are the agents leveraging like running code or searching for papers?
Yeah, so I'll show examples. They'll write their own code. So we don't write any of the code for them. So they essentially execute and write all of the code, and they can also read papers. Great. Cool. So maybe just a little bit more detail of how these group meetings happen. So in a virtual lab group meeting, right? So usually each meeting would have some sort of agenda, like a set of questions that the agent wants to answer. Then that's set up by the professor, right? Agent that says, okay, so the goal of this meeting is to answer this set of problems. And then all of the agents will participate, right? They actually go sequentially to provide their comments and ideas or updates that relates to that set of research questions. And they can do this in several cycles. So here's snippets from one of the virtual lab group meetings. It's actually one of the earlier meetings, right? So you can see here that basically the PI, the professor, initially just sets out that here's the goal of this overall project as we want to design some sort of binders to the recent COVID variants. And the reason why that's important is because if you have good binders, that's how you can actually get good vaccines or good therapeutic target. And then the immunologist agent spoke up next and made a kind of already quite surprising recommendation that suggested to design nanobodies. Right. For the audience, so nanobodies are actually quite uncommon, surprising choice, right? So usually, if you want to design proteins that can bind to some target like COVID, people normally design what's called antibodies, which you find in most animals, or in many animals. But nanobodies is basically like a smaller cousin of antibody. You only find it in camels and a very few animals. Right? So it's already kind of a surprising choice to design nanobodies rather than antibodies that's made by the agents. But as we'll see, this actually turns out to be quite a wise decision. And then the machine learning specialist agents spoke up next and said, okay, it's actually a good idea from an AI perspective to design nanobodies rather than the larger antibodies because they are smaller, so it's computationally easier to model and easier to predict. Computational biologist agent says that, okay, in addition to using these AI algorithms to predict the structures, let's also use more physics-style molecular dynamics to also get more robust predictions. And then you can see here the critic agent spoke up and says, because there's not a lot of public data sets around these nanobodies, let's also be careful about overfitting these models to those existing data. Yeah.
Do you decide, who decides about the granularity of what the principal investigator sort of states as the problem?
Yeah. So we wanted to, it's a good question. So we wanted to make this quite flexible, right? So basically all that we tell the PI is that we want to design some sort of binders to COVID, right? But we don't tell the professor agent, you have to design nanobodies or you have to design antibodies or how you actually go about doing that problem, right? So sort of similar to a similar level of granularity as, if I'm communicating with maybe one of my experimental colleagues, right? And I don't know too much about that field, right? So I don't want to micromanage what they do. So I just say, okay, my high-level goal is this, and then they can figure out exactly the implementation, how to go about doing it.
So I'm interested in the order that it's chosen for these to talk. Is this sort of randomized that you guys choose, or is it something that somehow the model can choose to go in at some point to respond? Is the critic always at the end, or can you have the critic go in between different specialists?
Yeah, so it's also an interesting question. So here, the full meeting actually can go for many rounds, right? So after the critic speaks, and then there's like another round of discussions of the agents can participate. So in this particular meeting, here, we just set like an arbitrary order of how the agents speak. But we'll actually say that we actually run many parallel meetings for each of these. So in each of the parallel meetings, in principle, you can actually have a different order of how the agents participate.
And do you notice any difference?
So we do notice that there's some quite interesting social dynamics that evolve, right? So for example, some agents tend to speak more than others, and when they get into disagreements, and then they have to try to resolve those disagreements. And one nice thing is that these AI scientist agents, they're very polite when they disagree, much more polite than us, right? So they try to resolve their disagreements very nicely. Okay, so that's sort of the group meeting, and then the one-on-one meetings are where each of the individual agents, right, then will meet with the PI or meet with the critic agent to solve a specific task, which could be like writing a piece of code or solving a subproblem of this much broader enterprise. And I was mentioning is that one nice thing about these virtual agents is that scientists is that their meetings are much more efficient than our meetings, right? So each of their meetings maybe would take seconds to a minute. So in the time that we had our coffee break, they had already run hundreds of meetings. They also don't get tired. So basically, what we do is that for every decision that's made by the virtual lab, we actually run 5 meetings in parallel discussing the same topic. And similar to if you ask me the same question on 2 different days, I might give you 2 different answers. Each time you run these parallel meetings, the virtual lab also explores different ideas and come up with different ideas, different suggestions. Right, so then after the parallel meeting, it's actually the job of the PI agent to read the logs of all these parallel meetings and then reach like a consensus and then go to the next step. Yeah?
Do these agents have an estimate of their confidence in their opinions? So on what basis do they have this interaction, no matter how polite? But how substantive can it? To be substantive, they have to have a sense of how much they believe in whatever they are stating.
Yeah, that's a good question. So especially when some of these agents get into disagreements, right? They do try to provide rationales to basically some levels of confidence or explanation justifications for their arguments, right? Like we see that even to a little bit of extent here, in that this is already kind of, I mentioned, kind of a surprising suggestion, right, to design nanobodies rather than more common antibodies, right? And there's actually, it tries to provide some justification for that by saying that, okay, it's, these are smaller and that's why we think this is a good idea, right? So I think essentially a lot of that justification essentially are in some ways like supporting or quantifying the amount of confidence and behind each of these arguments made by the agents.
I just want to confirm my understanding of the specialists. So those, there's no like parameters that are being updated, right? It's just based on like prompting that, like a system prompt or something that defines each of these?
So I'll actually, I have a few slides I'll show of, so we could do this with just the base model and with specific instruction, but I'll show a few slides where we actually do fine tuning to through this virtual lab school to actually update the parameters of the agents, right, to enhance its capabilities, and that does make a difference. Okay.
So it seems to me when they are having a debate, it's kind of going to the question you were asking. The agreement is more statistical, not like, say, if you're doing a prove or verifier. Eventually, you have to do some computation to verify or give me a proof that I'm convinced. So if, say, immunologist or machine learning specialist is really pushing you, right, the agent may just back down because it's still at a statistical level trying to get from the data what it's doing, or are you guys having some grounding or verification that is then?
It's a good question. So in that sense, then, we, the human researcher, we provide the final verification when we actually do the wet lab experiments. Right? So basically, the agents will have these virtual lab meetings. They'll propose, here are the actual proteins for nanobodies that they think is the most promising. And then I'll show you we actually make those nanobodies and test them in the real world to say, do they actually work as drugs or vaccines? So we have those results to show that, to do basically the final verification for the agents. Maybe one more, and then I'll, yeah.
Trying to figure out what's the difference, say, if there's an Uber agent, like doing a sort of a mega reasoning, like with itself, thinking out loud, versus here you have basically categorized each categories of sort of a special knowledge as some kind of specialist, and then you debate among them. What's the difference versus I'm a Uber agent, I just thinking out loud.
So we've done, so we've done a lot of these comparisons of just having like a single agent that tried to do these. So empirically, we actually find much better performance when you have these multi-agent sort of discussion settings compared to just having like a single Uber agent. And I think one intuition for this is that if you have a single agent, first the depth of reasoning tends to be much more shallow than if you actually have these more rounds of the multi-agent discussions. And also with a single agent, it's much less common to have disagreements, right? And often some of the more deeper scientific reasoning happens through the disagreements from these different perspectives. Okay, but great questions. Also foreshadows some of the things I want to show in a bit. But here, just in case you're curious, I'm just showing essentially how often the different agents actually speak in these virtual lab meetings. Right, so basically the PI in this case speaks about 20% of the time. No, we, the human researchers, we don't talk very much. We speak about 1% of the time, right? And then the computational biologist in this case has the most to do, so speaks the most, about 28% of the time. And you can actually see the critic also contributes quite a bit because it wants to provide some feedback to every decision that's made by any of the other agents. In this case, right, the role of we, the human researchers, is mostly providing like the high-level directions, and then we also tell the agents how much budgets we have to do the real experiments, right? But actually, all the hands-on work is done by the AI scientists. So in this case, they write all the code. In this example, all the code is written by the machine learning specialist agents, right? And then they do their own deployments and so on. So through this iterative process, then of these group meetings and then individual meetings, right? Then after multiple rounds of that, then the agent starts to make relatively rapid progress towards this quite ambitious goal of designing new nanobodies, right? And here, I think the agents actually did something quite interesting, which is that they came up with like a new computational workflow which is somewhat different from any of the workflows we've seen in previous publications. So the first thing that agents, one of the earlier decisions made by the agents is that they decided they want to use a set of tools, right, to help them to make these, design these nanobodies. So they came up with these 3 tools that they wanted to use. One of them is ESM, which is like a large protein language model. So you basically give ESM like a list of amino acid sequences, and ESM will tell you how likely is this to form like a functional or stable protein. Right. And then the agents that use the AlphaFold multimer, basically what it uses this is to take the top candidate selected by ESM, feed that into AlphaFold to basically generate like a computationally predicted structure, right, of how that nanobody candidate binds to the COVID spike protein. Right, so that's will be the output of AlphaFold. And the agents decided that that's actually not that reliable by itself, right? Then decided to actually use a different method. It's more like a physics-style method called like Rosetta, which does more molecular dynamics to computationally relax that AlphaFold structure and then recompute the binding energy. Right, so then it actually gets like a relaxed structure with like a binding energy score. So the agents decided to actually, instead of just doing this loop once, we'll actually do this four times iteratively to continuously select. It's almost like a continue to select list to generate like a final list of the nanobody candidates. So what I would say is that each of the individual tools has been used in many of the previous papers, but the way that the agents actually put them together into this workflow is different from what we have seen before, and moreover, the AI scientists also have modified these tools in some quite interesting ways that's different from what people have done before. So as an example, I mentioned this ESM is a protein language model, right? So you give it an amino acid sequence, and you can actually generate some sort of like likelihood of perplexity scores. There are actually a bunch of different ways of how you can extract embeddings, right, from this protein language model to compute a useful like a kind of a score, like perplexity score. So the previous papers did it in one way. Then agents actually decided to do it in quite a different way that, in retrospect, also makes sense to us. That the agents did it something that's quite reasonable.
Follow up on this point, do you feel like throughout human evaluation of agent output, do you think you have enough resources to decide whether the generations of the agent are truthful, faithful, or not? Or they're just like not factual, fact-based, but hallucinations?
Yeah, so we do look over their shoulders and look at their meeting logs. And I would say most of the discussions and debates, and also especially the code that's written here, they are quite reasonable. There are sometimes, of course, mistakes, but those are actually often caught by themselves when they have these disagreements, especially when they have these individual meetings with the critic agents that actually catches a lot of the mistakes, and then it fixes those mistakes. But to that point, I mean, our ultimate human verification, as I mentioned before, is that everything here is done computationally, right? But ultimately, we need to make the real proteins, nanobodies, right? And to verify how that works, which is what we did next. Right, so here, the agents actually designed this set of nanobodies using their pipeline, and then we, with our experimental collaborators, we actually make these in the real world, right? We actually synthesize and make these proteins, and then test them in the lab to see how well do they actually work in terms of binding to these COVID variants.
Yeah, but wouldn't it be perhaps more efficient to catch the error that she's talking about prior to you actually deploying, making it and going through the expense of, testing it and all that? So in other words, can we catch the error early on?
I think if it's a very obvious error, like if it's like a bug in the code, we can catch that because the code will not execute, right? And those are actually mostly soft-caught by the agents or by the critic. I think the harder kinds of errors is that this is a real new research problem, right? We don't actually know what a good nanobody will look like. So it's hard for us to look at this and look at one of the structures and say, is this actually really going to work or not? Right? Because that's just really beyond our capabilities. Right? So that's where we said, okay, let's not bias the agents too much, right? So they design these nanobodies that we'll just actually just make them and then test them, get to the ground truth from nature.
So my worry is like, say, even from the beginning, if an agent looks at some research data, some for some, just a little bit of probability here and there, chooses just a different wording, right? Instead of selecting nanobody, selected just something else, or something along the line, agent when formulating the objective function, choose the coefficient slightly different than should have been, right? Those are very subtle things and will create kind of catastrophic failure when you actually try to do the validation at the end. So I feel like it's like there's no free lunch. You probably have to kind of, like, I don't know, but it seems like a very fragile system.
Well, part of that is that we do run multiple meetings in parallel, right? So that does actually gives a lot more robustness, right? In that you can actually explore a bunch of these ideas in parallel, generate a bunch of code in parallel, and actually look at the most robust consensus answers. But the other aspect of this is that, this is sort of real research, right? Like even when a human chemist is designing these nanobodies, they have to make often somewhat ad hoc decisions at every step, with perhaps even longer feedback cycles, right? So ultimately, we do this research, we will have to learn from, the agents will have to learn from their experience in this particular case. They can then improve that for the next iterations. Right. So here basically I'm showing you then the wet lab results, essentially testing like how good are the nanobodies designed by these AI scientists. Right. So let me parse these plots. Right. So these are two example structures of new nanobodies designed by the AI agents. Right. So these are not nanobodies or proteins that you see in nature in the wild. And in the bottom, each plot here is basically we're measuring how well does actually bind to the spike protein of recent COVID variants. Right? So spike protein is the most important part of the COVID that you want to target. So first we can look at the bottom red curve, right? That's the open red curve. So what that red curve shows is that if I take the existing nanobodies that people had previously designed, right, if I say use them to test how well do they bind to the recent COVID variants, it does not bind at all, right? So the y-axis shows the binding affinity and it's basically 0. And then this second red curve is where we take this nanobody that's been optimized now by the AI scientist and also measure their binding. Now you can actually see much more promising binding to the recent COVID variants. Now what these blue curves show that if I take the same nanobodies and see how well do they bind, not just to the recent COVID variants, but to the original Wuhan strain of COVID from 5 years ago, you can see that the nanobodies designed by the AI scientists actually shows even higher binding, where it certainly retains the binding, but shows even better binding to the original Wuhan strain. And the reason why that's scientifically, I think, quite impressive is that it's actually extremely hard to have a single protein or a single nanobody that can bind across a diverse spectrum of viruses. Right? Basically, every year the virus mutates quite a bit. That's why the vaccine, you have to keep getting a different vaccine every year. But if you could actually have like a single binder that can bind well across the continuous spectrum of virus, then this is sort of the holy grail because that sort of can potentially become like a more universal vaccine. Right. And certainly a lot more experiments needs to be done to optimize and verify these nanobodies. But these are at least some of the first examples that we have seen of like actual nanobodies that shows such a broad spectrum of binding across very diverse COVID strains.
Just because this is very far outside of my area, what is considered like a good score on the y-axis? Like
yeah.
You were just trying to come up with something and like, this is promising enough that I would want to continue along in researching this.
Yeah, so I would say this blue curve here, the open blue curve is basically what we consider a good score because these are previous nanobodies that people have, the pharma companies have optimized to bind to the Wuhan strain of the virus. Right. So if you can actually get close to that or even beat it, that's considered to be quite strong binding.
Thanks.
I have a practical question. How do you do this? Like you run an AI lab, but you also have like a real biology lab. How does it all work? That just seems like really cool that you can do this.
Yeah. So this is so my lab is purely the AI lab, and then we run the virtual lab, and then we work with our experimental collaborators, John Pack's group. We're at the Chan Zuckerberg BioHub. So I'm also an investigator at the CZ BioHub, and then they have a platform there that actually specializes in doing a lot of these physical experiments.
Did it take a lot to convince them to do this?
I think they were quite excited to try this, to see, to really see how well would the AI scientists work. Yeah.
And then what did they think before they did the experiments? Were they like positive that this would be a good idea, or when they saw what they had suggested?
Yeah, so they looked at, and then we give them these structures. They looked at it and say, well, this looked promising. Based on of their domain expertise, they look like pretty very reasonable structures, look stable. So let's give it a shot.
Was there any kind of like human baseline of like you just tell the people at the biohub, oh, can you try to do this task? And then
we didn't have that explicit counterfactual, but in some sense you can view these open curves as being like the comparison. I mean, these are the nanobodies that have been designed through the standard human pharmaceutical companies, right, which have invested millions and millions to design these potential nanobodies.
Sorry, the flat curve is the one where they...
Yeah, these open curves, right? The blue open curve and then the red open curve. Yeah. So those are the nanobodies designed by the human researchers. But I just also want to caution that I think there's still a lot more experiments that needs to be done, certainly before these are really considered to be viable, actual drugs or vaccines. And actually, what we're doing now is we give all of these results back to the virtual lab. Like, for example, we can give it back exactly these plots, and now it's actually have already optimized like a second round of the nanobodies on top of these results. Right after some additional group meetings, now we're making the second round. So I think we're actually seeing this continuous learning experience.
Sorry, before you did this round, did you do rounds of dry runs where you had these virtual lab, get together, discuss things, propose experiments, and have experts say, well, these are great ideas, or we've tried them, they're good, or we understand this domain well, these are not very good suggestions, please try harder.
Yeah, we didn't do that. So this is basically like the first real experiment that we tried. So certainly there's been many of the sequence of the virtual lab group meetings where they iteratively try to refine their ideas and to come up with there's been a bunch of meetings where they try to refine this workflow. After they refined it, then they had a bunch more meetings to try to execute this and write the code. So we provide some oversight through all of that process, but not like actual. We don't really have very hard feedback like this is going to work or this is not going to work. Right? So we do tell the virtual lab, here's my experimental budget. Right, I have this much money, right, to run this much experiments. They can't generate too many candidates.
So it's really more or less one-way communication from the virtual lab to the human expert.
I would say it's, maybe like 80-20. Right? So we tell the virtual lab, we give the virtual lab here's the initial agenda, very beginning of designing, going after COVID rather than something else. And then in the middle, we also give it some, we basically tell it mostly different constraints that we have, like our constraints on the experiments, or our constraints in terms of time or the budget, or some of our priors.
But you didn't consider sort of a more symbiotic relationship where it was co-designed by the human and
Yeah, we didn't do that here. And this is also partly reflected in how little we spoke in these meetings. So we participate about 1% of the time. But certainly in other setups, I imagine these more, maybe like a 50-50 interaction could be interesting. So also to mention a little bit of how we try to have these agents to continuously self-improve. Right? So in addition to the virtual lab, we also are creating what we call the virtual lab school, which is sort of like a school where the agents try to essentially design their own curriculum, how to teach themselves to become a better expert into that particular domain. So how the school works is that, we, the human researcher, we say, okay, here are some of the topics that we're interested in, right? In this case, it would be like, maybe like you want to be an expert in using AlphaFold, right? Or using like ESM, this protein language model. And then what happens is that then that agent, in this case, the computational biologist specialist agent, first will generate sort of queries that it thinks itself should be important to answer, right? These are sort of how it designs its own learning curriculum. And then from these queries, it actually then goes into PubMed Central, which is like a central repository of open source scientific papers. It essentially retrieves papers that are most relevant for these different queries it designs. And then it decides which of these papers maybe are the most relevant, right? From these relevant papers, it first summarizes everything that's in each of those papers. We found that summarization to be a useful, important step. And then it does basically fine-tuning on top of all these summarized papers, right? So the fine-tuning part here now actually involves the model now updating its own weights. Right? After they do the fine-tuning, then the agent will do like a self-assessment, right? They'll generate different QA questions around to that topic, and then test itself to see how well has it actually learned that particular topic. And you can imagine also doing this iteratively through many different rounds.
What is the motivation for summarizing the papers and then fine-tuning on the summaries as opposed to just fine-tuning on the papers themselves?
Yeah, so we did both, and we found the first thing we tried is actually just having the agents find the relevant papers and then fine-tune on the full papers. First thing is that ends up being a bit too noisy. It also has just have too many papers because here they're extracting potentially thousands of papers. So if you fine-tune on the full papers, there's actually a lot of potentially irrelevant information that ends up distracting the agents. We actually found just empirically that first having the agents read each paper, summarize it, extract the parts that are most relevant for that topic, and then fine-tune itself on the summary to be more effective. And these are just some of the results of for these different topics, right? On these sort of like QA evaluations, right? You can see that you can get pretty consistent improvements, but from the agents actually going through this own self-learning, self-improvement curriculum. Especially on some topics, like learning about these more recent COVID variants, there's actually quite a substantial improvement in the ability of the agents to reason about that topic. Yeah.
But even the baselines without fine-tuning still seems really high.
For some of them, yeah.
So even without updating the parameters, just with the right kind of prompting, you can get this
you can get pretty far.
Setup. Wow.
Yeah. I will show some failure cases of the baseline models, though. For example, we take the original agent without fine-tuning, and KP3 is basically one of the more recent COVID variants that started to spread last year, right? So it's actually a little bit outside of the training data of these models. And then you can see, okay, if there's some questions, right, related to some mutations that KP3 variant, then the original agent is actually really essentially not able to answer that or sort of give like a fluff answer, right? But then after the fine-tuning, the agent is actually able to provide very specific answers about what are the mutations in these recent variants.
So the previous slide, I'm curious if you omit a few of those blocks, modules in your scales of analysis, how it impacts the performance. In other words, what's the contribution of each of those scales, each of those components in the performance? So you can have a one-to-one.
Yeah, that's something we're actually doing now to do these more systematic oblations.
I see.
A follow-up on that question, is the design of the protein contributed by the AlphaFold algorithm or the agent system? I'm just clarifying this thing.
Yeah. So the agent designed this workflow, which uses AlphaFold as one of the 3 tools, right? But the design of protein is actually not just from AlphaFold, but it actually comes from running all 3 iterations. So it takes AlphaFold, predicts structures, actually runs like a physics simulator to relax that structure, right, which is called Rosetta, and then iterate through this multiple times to generate the final structures. So the agent basically designed this workflow and extends each of the individual tools, right, to design this process. It's pretty actually pretty interesting how they did it. Okay, so that's what I want to say about the virtual lab. I mean, our vision was this: is that if you think about the scientific research process, right, from start to finish, there are many different expertise that are often needed in different parts of these research projects. And our goal is that with the virtual lab school can actually have a very scalable way to create a pretty diverse set of AI scientists with different expertise, right? Each of them will have a deep expertise in a particular domain, right? And then for different problems that people arise, right, in drug discovery and development, different expertise are needed. Then we can actually quickly assemble, right, these different sub-teams, task forces, right, with the relevant expertise to solve each of those problems. Okay. So in the second half, I want to also share, I related the project we did of now, instead of having the AI do the research part itself, we want to have the AI agents to help us with the scientific review process, right?
Yeah. Can I just ask you a quick question before you move on? So did you actually scale up to many types of experts, or is this something for the future? And related to that, do you have any thoughts about, how this, how these how the system would know, which experts to leave out, or, are they getting in the way? You really want to drill down to fewer, etc. So.
Yeah. So we are scaling up the designing these experts. So we have this virtual lab school, which is like a generalizable recipe for how to create each of these agents. Now we're actually doing this for a broader list of agents with different expertise now. So I think currently we have maybe like 6 or 7 scientist agents. Now we're actually scaling that up. The team selection part I think is quite interesting. We want to do more experiments there. We've tried running these experiments with basically teams of different sizes, and there is sort of like a sweet spot. If you have like too many agents in these group meetings, then they sort of distract each other, and it's not that effective. There's also quite an interesting component about the personalities of the agents, which we're also doing experiments, right? I mentioned before that a lot of these agents are actually quite polite, but we've done some experiments where we actually try out different personalities. For example, you can have maybe like a very neurotic critic, right? Like the George Costanza of the reviewers. And there are some settings where actually having that more neurotic critic is actually quite helpful, right? But you don't want to have everybody being like a George Costanza style, right? So there's actually a lot of, I think, almost like, I view this virtual lab also being like also a platform where we can actually do some quite interesting social science of how to design optimal teams of researchers. Yeah.
Just on an implementation level, I'm curious how you treated like memory and context throughout the research project. Is it just like a longer and longer context window, or do you do like retrieval augmented generation?
Yeah. So we have this memory mechanism where we do store all of the meeting notes from the previous meetings, and then we periodically basically have the professor agent to summarize that into like a list of to-dos and next steps. Basically says, okay, so here's everything we discussed so far, and then here are like the concrete to-dos, right? So having that periodic compression is quite useful for these.
If I step back, like in some sense it seems like too good to be true. Because it's like, I know of other evaluations where agents really struggle with like long horizon tasks and even just like more concrete things like software engineering type tasks that are more well specified. And here it sounds like you're saying these agents did much better than like the people who spend millions of dollars trying to research the same thing. And so, like, is that also the impression that you have, or is your reaction to this different?
I think there still needs to be a lot of work to be done to make this like really work well across many different settings. Right? I think in some sense, that's why I mentioned in the beginning, actually, like the design, the choice made by the agents to design nanobodies ends up being quite a fortuitous decision for them, right? Because that's something that's actually relatively well controlled within those agents. It's also something that with these external tools like AlphaFold, you can actually get quite reliable, not perfect, but pretty good estimations of the structures of these nanobodies. If they had actually decided to work on a different strategy where they do not have these very good tools, right? I think they would have had a lot more struggles with designing good candidates. Maybe in the interest of time, I'll move forward a little bit, but okay, I'll take one more and then I'll move forward. Yeah. We tried running some of these different oblations in parallel, where for example, we say, okay, like if I have just the generic GPT-4 versus one of our more specialized agents, right, participating in one of the group meetings, what happens. It turns out that the generic model often makes a lot more mistakes than our specialized agents. But we haven't run the full process end to end many different times. So partly it's because the experimental part from the human experiments is the bottleneck. We can only basically do that once. Right? It takes about 6 weeks to actually make these nanobodies. It's actually pretty expensive. So we're not able to actually do a lot of that in parallel. Okay, great. So then second, in maybe the last 10 minutes, I want to share like how we are using these also teams of these AI agents to help us with a related but quite different problem, which is helping human reviewers. And the motivation for this is that for a lot of the AI conferences, we're just seeing this rapid increase, right, in the number of submissions. Like, for example, iClear has basically been like doubling in the number of papers they're getting, and we know that it's getting hard to find high-quality reviewers and for all of these papers. So motivated by this, we actually had a collaboration with iClear, which is one of the main AI conferences this past year. Basically, we designed an AI agent to help iClear reviewers, right, which was deployed on around 20,000 iClear reviews, which just finished. The iClear decisions were just made last week. So I'll just tell you hot off the press what are the results of having these AI agents helping with these iClear reviews. Right? So iClear basically had 40,000 reviews they had to handle. We did a randomized A/B test. About half of them, 20K, had help from our agents. 20K did not. So we will look at the effects of these agents. So here in this setup, we explicitly designed our agents to not review the paper by itself. So the setup here is that they are strictly meant to be an assistant to the human reviewers. Right. So, and this is sort of like a co-design decision we made with iClear. So the setup here is that there will be a paper submitted, right? So one of the human reviewers, right, will generate like an initial draft review for iClear, and then our agent will actually look at the original paper submission and also the original review and provide some sort of comments to the reviewer on how they might potentially improve that review. Right? And in particular, we looked through a lot of the previous papers to see what are the basically common pitfalls or common limitations of previous reviews, and to generate like a checklist for the agents to look over. And so the agents, for example, will look for, okay, this original review, maybe did it overlook some relevant information from the paper? If it does, then we'll try to provide some feedback, right? Did the review provide some criticisms that are fairly vague, and maybe perhaps lack justification? It identifies those, then we'll actually also provide some feedback comments to reviews. Or if the review had some like more personal or unprofessional comments, the agent is also meant to catch those and provide feedback to the reviewer. And I also say that this work is led by Nitya, Mert, and Jake, who are fantastic PhD students in my group. Yeah.
The reviewers know that this is an AI giving them feedback.
Yeah.
Okay.
Yeah. And here are just some examples of the kinds of feedback, right? Maybe like the original comment could be, this paper could use more experiments, and that's maybe a little bit vague and can be a bit frustrating to the authors. They don't know what the reviewer is actually asking for. Then the feedback that the agent will provide to the reviewer would be, okay, can you make this more specific by providing, let's say, specific methods you wanted the authors to compare with, right? Specific baselines. Maybe the original review had some comments that like, okay, yeah, this paper is missing some experiments, but perhaps the reviewer actually missed some part of the paper, or maybe the part of the supplement, right? So the agent actually will provide a feedback like, okay, maybe you talked about this transformer experiment, but figure 5 of the paper actually explicitly have results for transformers, right? Maybe take a look at that again. Or if the review has some, let's say, perhaps unprofessional comments, right? Then the agents also try to provide some feedback to that. And this is all optional feedback, right? So the reviewer can perfectly ignore any of the feedback from the agents. Right. So it's it's all completely optional for the reviewers. So how the feedback is given in the conference is that the agent will actually provide this one-time feedback on these reviews, right? And then review can have the option of they could ignore the feedback. That's perfectly fine. many of them do. But if they find the feedback to be useful, they would they have time to basically update their review before those reviews are made public to available to the authors. Right. And then the rest of the ICLR process is exactly the same as before. Okay. I'll be fairly brief here, but under the hood is actually multiple language models working together to provide this feedback. So essentially, we have like 2 cloud models that we've given a lot of in-context learning examples from previous reviews, right, to basically look for what are some good, useful, constructive criticisms. And they'll basically provide initial feedback, which is an aggregated, and then there's a self-improvement aspect with the critic, and then it generates the final feedback to the reviewers. But the overall goal here is that we don't want to necessarily change the review very much, right? This is really meant as an assistant to help hopefully make the review more actionable and more informative for the authors. Okay, so in this ICLR conference, so altogether this year, right, ICLR received about 44,000. They received over 10,000 papers, and they had to get 44,000 reviews for all of these papers. Right? So it's a huge number. And then this is our A/B test. So basically, for half of these reviews, were randomly selected to receive this AI agent feedback. And then about 27% of the reviews that received the feedback actually decided to update their review based on the feedback to incorporate actually some elements of the feedback. And the rest of them, maybe they ignored the review, or maybe they looked at the review, decided it's not that useful. Right, so we don't know exactly what happened with the rest, but this 27% is the ones that we're most interested in to look at. So this is actually kind of interesting. This is basically looking at, the top row looks at basically the number of reviews that are submitted as a function of time, right? So this red curve is basically the deadline for submitting reviews. You can see that, okay, there's as you expect, there's a big spike exactly when the deadline is, and then also a bunch of reviews that came after the deadline. And then this bottom curve basically shows essentially what fraction of these reviews actually then would incorporate the AI feedback, right? You can also see this pattern. I mean, the baseline, the steady state is about 27% on average, but it ends up being higher for the earlier reviews and then drops for the later reviews. I think that makes sense because the reviewers get busier, and then they have less time to incorporate this AI feedback.
Do you have an idea if agent feedback on average changed the reviewers, actually led to more positive or negative reviews?
I'll show those results, yeah. So here's some example of how the review feedback is actually incorporated. Right, like this would be like an original comment from one of the reviews. Right, there's some assumptions about interpolation. It's somewhat vague. Right, then the feedback to the reviewer in this case would be, can you actually give some specific examples, right, of why this interpolation assumption you think is particularly necessary or unnecessary, right? And then after this feedback, this is the modified review, ends up being, I think, actually very informative. It says, okay, so there's some previous papers about this algorithm, gives a citation that says that they considered this non-interpolation regime and asked basically the authors to essentially do a similar analysis, right? So this ends up being, I think, a much more informative review compared to the previous comments. Here's a second example. The reviewer just had something like, what's the performance on Llama? The feedback says, oh, there's actually a table that had the results on Llama 2. And then the modified review actually says, okay, yeah, so, okay, now you look at Llama 3. Maybe a little bit more useful for the authors. Okay, so basically, we provided around these 18,000 feedback comments. Among those, about 2/3 of these comments provided by agents were actually incorporated when the reviews get updated. And as a result of that, the length of these reviews actually increased by about 600 characters. So they're actually becoming more informative. Usually, when they incorporate these comments, the review gets longer and more informative. Okay, and this basically just shows the x-axis is essentially the number of feedback the agents provides. Right? Sometimes it's one feedback comment, could be 6, sometimes more. And then the y-axis is that how many of these feedback comments are actually incorporated. So basically, the diagonal are the situations where every comment provided by the agents actually ends up being incorporated by the reviewer when they update the review. Right? So like in this group here, like the agents provided 8 comments, 8 suggestions, and then 08 are incorporated in the review, which is actually quite high. So you actually do see a pretty big concentration around the diagonal, which is, I think, quite interesting. So these gives us some proxy, right, that the reviewers are finding these feedback comments sufficiently useful that they actually take the time to go back to update the review and explicitly incorporate these comments.
Question. How do you, while you're talking about how to evaluate this, there's a story about the supervisor who says after getting a report, you can do better. And after 3 rounds of this, over which the report improves, he says, okay, now I can finally read the report. So how do you know that, how can you compare what happened to sort of just a simple constant or very simple heuristic that just says, go back and check your work again.
Yeah.
Give me a better report. How would they do?
Yeah, it's a good question. I guess one of the things we try to do here is that when we look at the incorporation, right, we're looking very specifically, say, is this specific suggestion actually incorporated versus like a generic change to the review, right? And here we're saying, yeah, like in this case, this specific feedback provided by agent is actually explicitly incorporated, right? So at that point, at least we're looking at, are these specific comments incorporated rather than just like a generic, just improve your review, yeah. Maybe I'll just wrap up and then we can take any further questions. Okay. And then, so the final thing I want to show, which is basically how does this actually affect the review scores. So basically with iClear, what happens is that there's an actual overall rating, right? And then which is ultimately what matters, right? And then there's also, provides like a sub-rating for different aspects of the paper, like contribution or presentation quality. The control group are basically the half of the papers that just had the regular workflow, didn't get any AI feedback. And then the treatment arm is basically the feedback group. Right, so here we do see, it's a small increase, but statistically significant change in that the papers in the feedback arm had overall like a bigger boost in their ratings if you compare the pre-rebuttal score versus the post-rebuttal score. Right. Our interpretation for this is that because the feedback comments made to the review more actionable, right, then the authors are more able to engage directly with what the reviewers wanted to say, right? So that actually led to potentially like a more engaged interactions and hence like a bigger change, bigger delta in the overall ratings. At least I think, yeah. So we're still digging into the data and more, but I think that's our initial assessments. So just to wrap up here, I think, again, this is sort of the first really large-scale experiment of using these AI agents as a part of the peer review process in like a real journal or conference. At least what we find here is that a large number of the feedback provided to the agents, even though these are optional feedback, they are incorporated explicitly by the reviewers. So at least somebody found them perhaps useful to incorporate. And we have some evidence that these feedback from the AI agents does enhance sort of the author-reviewer engagement and potentially also improves like the outcome. But we need to, we're still digging into those analysis results more. Okay. So I'll just wrap up here. And we want to give these 2 examples with the virtual lab and the feedback agents to sort of illustrate, quite early steps of why I'm quite excited about the ability of these AI agents with appropriate optimization and guardrails and training to really help us across the scientific discovery process, all the way from designing experiments, carrying out these experiments, all the way to more the later stages of helping us to improve our peer review process. So the first virtual lab is led by my student Kyle Swanson in collaboration with our experimental partners, Jiang Pak's group at the BioHub, and then the feedback agents led by my students Nitya, Mert, and Jake in collaboration with the program committees from iClear. Yeah, so happy to take any further questions.
Hi, James. Great work. So I'm thinking, like, right now, the virtual lab work, it's just prompting GPT-4o, right? I checked the code. So
yeah.
So these models doesn't have the reasoning capability. So do you think it would be better if the next version will be really sort of leverage these reasoning models? So I'm not sure how useful each of these, like immunologists, these virtual specialists, is that useful? Because it's prompting anyway. So you can just train a reasoning agent that's, we can get rid of all these different agents because these are reasoning agents and they're capable of multiple step tasks. So they could do the whole thing. And
yeah, so I think it's interesting. We are doing experiments now. I mean, we can swap out each of the individual language models with like R1 or O1, O3, and we're doing those experiments now, and we're also fine-tuning those reasoning models. We're fine-tuning the O1 models to have more specialized expertise. So we do find certainly there are benefits to using the reasoning models as the base of the agents. We also do find that this multi-agents setup, even with reasoning models, is still much more effective than having a single agent setting, especially when we further specialize and optimize each of the individual agents. Right? So we're doing experiments we have done experiments where we've optimized fine-tuned O1 models, right? And then those actually have much deeper expertise.
So I think my question is, I think that's great, but I think these reasoning models are still not agent models. So like we can actually train these models to be able to interact with the environment, perform actions. I think would that be another?
Yeah, I think here I agree. I think here a big part of this is that the models need to use these quite sophisticated tools, right? They have to use AlphaFold, but not just use it off the shelf, but they modify it in interesting ways. Right? So I think that's actually a part where some of these virtual lab school type setups where the agents then becomes more of like a super user for those tools is actually quite useful. Yeah, so even just the off-the-shelf reasoning models are not very good at using these tools.
Right. We should probably do only one more question.
Okay. Maybe we can do one here?
Sure. You mentioned you use fine-tuning to specialize those agents. Did you look at retrieval augmented generation as an alternative? Do you have a thought about the relative goodness of those two approaches?
Yeah. So the actual, for example, the current optimization we have actually does both, right? In that the agents, it designs its own curriculum by retrieving the relevant papers. It retrieves hundreds of relevant papers and summarizes those papers, right? So there is that RAG component already. And then we do find that if we actually explicitly do fine-tunings of the agents on top of those retrieved documents, that also gives additional improvements. But yeah, happy to discuss more during the coffee break.
That's what I was going to ask. And I feel like because OpenAI also basically uses the kind of